you everyone for coming out this morning, and thank you for the invitation. This is work that was published as part of the subcortex special issue in Neuroimage in 2022, I believe. And what I'm going to do is zoom in and just give you an overview and try to build an intuition on how we can use this concept of edge time series and edge communities to study interactions in the brain. Now, we all know, I'm sure we all have an appreciation for the subcortex and the subcortical structures in the brain. Otherwise, we would have chosen to sleep in this morning. And it is extensively interconnected with the cortex, and it is made up of several nuclei. I today will focus on the striatum, the thalamus, as well as hippocampus and amygdala, kind of the main structures that are that we can image with pretty good SNR. And then we know from a lot of work by Suzanne Haber and others that there is a topographic organization of the structural connectivity that's projecting between the striatum and the cortex. And we know that there's functional organization is there. And there I'm pointing out the Melbourne subcortical atlas done by Etienne and others. And it's the one that I'll be using here in this work. So briefly about the edge time series. In conventional functional connectivity, when we do a Pearson correlation, we're examining a bivariate relationship between two regions. With edge time series now, we look at the connections. So in terms of Pearson correlation, if you're familiar with it, we just stop one step short of averaging the z-score, the element-wise product of the z-score time series, and we end up with an edge time series. So now we have a time course for every connection. So here you can see IJ, IL, and JL. And what it allows us now to do is to perform clustering or modularity, where instead of an assignment of a community to a region, we get a community assignment to an edge or a node. And we can bring that back into a matrix, and you can see this is a 7-cluster solution that was derived from the HCP data, the 100 unrelated subjects. And when I thought about this, I'm like, oh, cool. Now I can look at what sort of unique motifs exist there. So in the network neuroscience, we're really familiar with structural motifs, so these unique smallest elements that we can study, typically a triangle. But now what we can do is look at community motifs. So we can have a closed loop where all 3 regions are in the same community. I completely made these names up, so if they don't make sense to you, I'm open to suggestions. We have a forked one, so this is with respect to that node L. And then you have an L shape and diverse where you have 3 communities. So these are the unique ways that you can organize that these triads can appear. And just for me, for interpretation, and I think what's useful here is to always have an anchor node or a reference node. So in this case, it's L. So we're looking at how the time courses or how the dynamics between node L and any other 2 nodes are behaving based on the edge community structure. So I mentioned this, again, showing this matrix that was on the previous slide, it's highlighting that there is organization in edge community structure, and it's really pointing out this dichotomy between among the resting state network, but also subdividing it into more the primary association networks and then heteromodal higher order networks. There's 232 nodes there, so what you're seeing at is the EO7 networks organized on the rows and columns. And if I show this a different way, where now I look at it from a node-wise perspective, and I say, well, how many edges from a particular community are touching that node, you can see there's pervasive overlap throughout the cortex. And that not all edge communities have a component in it or have edges that are touching the subcortex. So there are edge communities that are purely cortical in this scenario, and there are others that have a communication between the cortex and the subcortex. And as I mentioned, that there's this separation between heteromodal association systems and the primary visual somatomotor systems. So if we summarize the edge along rows, edge community structure as entropy, so how much variability there is in the community assignments, we see this profile kind of emerge where you have the control and default mode systems kind of hanging around 0.5, and then you have a little bit of higher entropy in the primary systems, and then the subcortex is a little lower, but there is some distinction between hippocampus, amygdala, and then the core striatum and thalamus there. A third way to look at the data, because my postdoctoral and graduate advisors have always taught me, look at it from as many angles as possible. So now we're projecting it onto the brain. So it's the same matrix. Now I'm showing you what it looks like on the cortex and on the subcortex. So now focusing on the edge communities, focusing on the edge communities that have a subcortical component, we can see community 1 is really, its edges are widely distributed throughout the striatum. And then edge community 3 is mostly restricted to the hippocampus, while edge community 4 is more restricted to the predominance of its edges have a connection within the striatum. And then we have 5, which is another one more hippocampus, amygdala, and posterior thalamus there. So there is variability among the edge communities in terms of the communication similarity there that we're estimating from edge communities. And this is where I was really intrigued and interested in looking at these interactions. So I decided to, instead of taking the whole matrix, which is a lot of information, I wanted to focus just on the cortico-subcortical interactions. So we pull out this subgraph, which only looks at the edges between the subcortex to the cortex. And from this interaction block, we can now compute metrics either along rows or along columns. And we get insight about whether it's subcortex or cortex, or cortical information that we're looking at. So now looking at the cortical nodes and the entropy of the edge communities in the cortical nodes, just from this interaction block, we still see that similar pattern emerging, where the heteromodal systems have a lower entropy relative to primary systems. It's just it's just lower. You can see it's sliding down lower on the y-axis, and it's because we have fewer communities in this interaction block that we're dealing with. And then again, the similar pattern is emerging in the subcortex. We can look at how similar the edge community distributions are between two regions. So we take the rows of this matrix here, and we compute the similarity among all rows, so we derive a new similarity matrix. And if we do it for the entire network, so including the cortical, this part, the cortical nodes, as well as the within subcortex part, you can see that it looks like this. But if we only take the edges from the interaction block and compute the similarity matrix, they're R1. They're essentially identical. So you can estimate the similarity of edge community structure solely from the interactions between the cortex and the subcortex without the full network. And just to complete this, if we now look at the cortical systems now, and we look at the entire graph, and we compute the similarity versus only computing similarity from the interactions between the cortex and the subcortex, they are, again, highly correlated. So both entropies and profile similarities that we estimate by looking only in the interactions between the cortex and the subcortex are very much similar to the full network. What I've shown you up to this point has been a consensus solution for the 92 unrelated subjects. So just for thoroughness, I wanted to make sure that I could see something like this in the individual subjects. Now, this is a computationally intense process to do the iterative edge clustering, which is why I first restricted it to 92 subjects. But what we can do with the clustering procedure is give it initial seeds. And what we did is we used this consensus solution that I have shown you up to this point as the starting point. And now comparing the similarity through normalized mutual information of the solutions that we get, either from all participant to participant comparisons or all to the consensus partition that we used as a starting point, there's still inter-individual variability in there. So I wanted to make sure that by giving it these initial seeds to the clustering algorithm, that I wasn't biasing it, that I was still capturing variability in there. And that is indeed the case. So it is a viable method, at least in my opinion, to reduce on the computation time and be able to do this at the single subject level. And again, computing the entropy of the edge community solutions for each participant in the HCP 100 unrelated cohort, we get that similar pattern with now with the variability in there where each point is a single participant. When we look at the entropy, I'm focusing in of looking at entropy of edge community labels either within the primary systems, which I refer to as the visual, the somatomotor, and the attentional systems, or the heteromotor systems, so the limbic, frontoparietal default mode, and then the subcortical. We can see that the entropy is highest in the primary systems. Going on to the next is the heteromotor systems, and then the subcortical. So the primary systems actually have the greatest diversity or the greatest variability in the edge community structure. They have more edge communities that they participate in a stepwise manner than the heteromotor, than the subcortical. And then now examining the same, but now looking at those similarity profiles of edge community structure. Within the, so let me show you, focusing on the left part here first, this is looking at similarity of nodal edge community assignments within the respective system groups. So this is within the primary systems. You can see they're similar to each other. Nodes within the heteromotor systems have a lower similarity to each other, so they're more variable within that group. And then the subcortical is pretty much on par with the primary. And then stepping outside kind of the main diagonal of that matrix where we looked within the systems, now looking at the blocks between systems, we can see that edge community similarities primary to heteromotor are actually the lowest. So they have the most variability. Those interactions between primary and heteromotor systems are the most variable, followed by the primary to subcortical, and then heteromotor systems, the high order systems to the subcortical. The similarity of those profiles was the highest. Finally, on to motifs. So when we did permutations testing to see is there any particular motif that occurs at a greater probability than we would expect by chance, this fork with respect to a reference node R in this case, which is a subcortical node, and then any two other cortical nodes. We can see that the fork triads were significantly overrepresented in the striatal and thalamic structures, while actually this L-shaped triad was underrepresented, significantly underrepresented. Breaking this down by community, you see the edge community 1, which was widely distributed throughout the striatum, and thalamus has significant overrepresentation of fork triads, and edge communities that were predominant in the hippocampus of amygdala, they also have that significant representation at the edge community level. They just didn't show up here because it's largely dominated by these two communities. Just to do some checking then to make sure what do these triads look like. We looked at whether or not there was some sort of bias in laterality of these triads. So is it a left subcortical node and two cortical nodes on the left side, a left or right hemisphere, or contralateral to the subcortical node? You can see these triads are actually pretty evenly distributed. So when we're examining these fork triads, they're not just restricted to living within the same hemisphere or contralaterally, they're distributed throughout. And yes, so this is the same, this is divided up by these three communities that show the significant overrepresentation of the fork triads. And then finally, looking at any sort of distance relationships between the two cortical nodes. So are these fork triads that are significantly overrepresented? Are these just two adjacent cortical nodes or two cortical nodes that are close together in space? And this is colored by edge community. You can see there is a distribution. Some are close, but there is a distribution there. So it's not just regions in a cortex or nodes in a cortex that are together or very close to each other in space that are being represented here in this fork triads that are in a brain. So to summarize, I think overall edge community structure, we saw that this distinction between attentional and higher motor systems, which is something that's consistently been shown not only in edge time series, edge community structure literature, but also in functional connectivity literature overall, this separation. The functional organization of the subcortical regions, there's no directionality here, but based on what I've seen of the ability to estimate the similarity profiles solely on the interactions, I think there is influence of whether or not we're only looking at the cortex, we only have a graph for the cortex and not the subcortex. The contributing signal is still in there. We just didn't measure it. So there is influence, or they are influenced. This overrepresentation of forked cortico-subcortical motifs, this is consistent, at least in my thought, with the anatomical roles that we see for the subcortex as regions that have a lot of feed-forward feedback loops going between the cortex and the subcortex, where they're synchronizing information, where they're synchronizing regions, and integrating information as well. And with that, as it was noted by Boris in the beginning, I'm starting at the IU School of Medicine, so I just wanted to give thanks to my postdoctoral mentor, Olaf Sporns, who's actually at NetSci, accepting a well-deserved Euler Award this year. I really appreciate him for putting up with a clinical research scientist for 5 years and letting him hang out in his network neuroscience lab. Big thanks to the rest of the Sporns lab, Richard Betzel, Brain Networks and Behavior Lab, and then the IU Network Science Institute and the NIA for funding the duration of my postdoc. Thank you.
Hi, we're open for questions. I would encourage people to come to the mic and ask questions if you have any. Are there questions?
Otherwise, I have a question maybe.
Sure.
Terrific talk, thank you. So systematic variations in SNR across cortical as well as subcortical regions, is that something that's important to consider for variability in edge community structure?
That is an excellent question. Yeah, so I think it is an important consideration, especially just the ability to accurately measure the subcortex and overcoming that SNR. I didn't share, it's too early, some of the work, but step one was for me to do this in the high quality data, right? There's nothing like HCP, it's high quality, but I'm now moving on to trying to apply this in Alzheimer's disease patients and clinical data set. So there, those edge community matrices, those images, they're not as pretty. They're noisier. So I'm putting some thought into consideration of how to best get the signal and kind of reduce the noise. But I think, yeah, you need to consider the SNR, especially the sequence and the system that you're doing imaging on.
And that was with the 3T or with the 7T data set or both?
So this was the 3T Connectome data set scanner. Yes.
Hey, this was really cool. Thanks. I tried to put a question in the chat, but I don't know if I did it correctly or not. In terms of actual neural connections, when I think about the brain, primary sensory regions really only connect to their respective association area, right? There's not connections from say, motor cortex to the rest of the brain. It really only goes to motor association, and then that connects on, whereas the hetero and transmodal areas connect throughout the brain. Your data seems to say that at least in functional connectivity, not network connections, it's the opposite, right? That the primary sensory regions are more connected to other networks, or at least co-vary more with other networks, and the heteromodal regions don't. How do you kind of reconcile that?
Yes, so this is a tricky kind of thought exercise, right? So the functional connectivity, and especially the edge community structure, we're just looking at similarity of time courses now. And in the edges, we're now looking at, instead of two regions, how pairs of regions go together. So it's this higher order relationship. So it's not just, one node to another. It could be how similar are two nodes versus any other two nodes. So there's from a functional perspective, there are other nodes in there, there are other regions in there that can have mediating effects through indirect connectivity that functional connectivity just doesn't capture. Yeah, that would be my best answer, best guess that I can give you. Yeah.
It's hard to kind of reconcile it, but it makes sense that functional connectivity doesn't get at direct connections per se. It's things that are kind of higher.
Exactly, yeah.
All right, so now